So we will have our next speaker, Pratyay Sengupta from IIT Madras. So it is an early career talk. So the title of his talk is Microbial Interaction Network Reveals Co-occurrence Patterns Across Built Environment Microbiomes. So you have 20 minutes time, okay?
Hello, so good morning everyone. And I am Pratyay Sengupta, a PhD scholar in the Department of Biotechnology at IIT Madras, working with Professor Karthik Raman. And I still remember I visited this hall 5 years back during our INSA summer fellowship program. And I'm very excited to give a talk here after 5 years almost. And I thank the organizers for giving me this wonderful opportunity to present our work. So in next 15 minutes or so, I am going to present my work on recent work on identifying microbial co-occurrence patterns in different built-in environments. So let's start with what is a built environment. So built environment is essentially everything that surrounds us, everything that is built by human, for human, and that is used for regular human activities. And as we are the habitat of these environments, our microbiome is deeply influenced by these built environments. Mostly because number one, these built environments act as a unique reservoir of different microbes, especially our human associated microbes. And it also works as a barrier for our exposure to natural environments, and it restricts the transmission of different microbes, good microbes to our body. It also reduces the microbial diversity and accelerate different diseases. So why it is important to study built environment microbiome? Because as we know, in the post-industrial revolutionary era, built environments has changed drastically, and it has an impact on a lot of aspects, and one of the major thing is public health. And today's built environment has exposed us to multiple sort of pollutants and toxins, cause lots of allergy and asthma. Not only like that, our regular home environments are also harbor different gut microbiome. It basically decide how gut microbiome going to colonize in infants and determines its subsequent colonization in the later stages. And also in more specifically in hospital and other associated built environments, the microbiome cause different healthcare associated infections. So to address that space, we tried to come up with several questions. Our first question is, this microbial, what are the microbial ecology or microbial diversity in different built environments, if there are at all any differences or not? And second, if there is a difference, how these different built environment affect that particular interaction, and if certain interactions are affected by environment, is there any pattern that we can find in terms of cross-spreading across these communities? And to address these questions, I choose a paper by Dr. Niranjan's group, and he made my work easy here by his talk yesterday. So I'll just go over the data what we had. So the paper had the data from nearly 438 samples from a hospital, Tan Tock Seng Hospital in Singapore, and it has subsequently data from office space, which is like our home or office space, particularly from the Genome Institute, and also we utilized nearly 100 samples from metro station in Singapore from the Metasub initiative. So our first, as we were particularly interested in bacteria and bacterial interactions, so we tried to see what is the bacterial composition, and very known, nothing surprising here, the bacteria are the most prevalent in all these environments. But very interestingly, hospital had a lot of viruses here, nearly 10 or 15% here, and that is there also. So not always bacteria are there. And then followed by, we tried to see, are these environments diverse enough? And yes, alpha diversity showed that hospital is much more diverse comparatively to metro station, which is somewhere in between, and office is very low. And also beta diversity, not much on NMDS axis 1 we are seeing, but beta diversity on NMDS axis 2 showed that similar kind of pattern, where hospital is much enriched, whereas office and Metasub in between. So this specifically showed us that these environments, built-in environments, microbiomes are diverse and very unique to them. And as a class level agglomerate, you can see there are three particular classes like bacilli, gamma proteobacteria and Clostridia, which are popularly known for harboring nosocomial pathogen. So those who are not aware, nosocomial pathogen are the pathogen which usually stays in hospital and affect the immunocompromised patient, and they are not much present in other environments as such, and not at all in the office, and office has a lot of alpha proteobacteria which is we see in soil and other environments. So that shows us that these environments are very diverse. Now we try to see if these are diverse, what are the microbial interactions in these particular places. So for that we took microbial relative abundance and put some cutoff to ensure that they are well abundant as well as prevalent in these particular environments. Then before generating a network, we pre-processed the data. We imputed zero value using a small value delta using a non-parametric function and multiplicative, that's called multiplicative imputation, followed by a normalization using total sum scaling, and then as this is a relative abundance data, we use a G-CODA method. So CODA here is a very popular term in microbial because of relative abundance. So it's called conditional dependency analysis, and G-CODA is a tool that specifically uses a graph-based approach, and then it works very well with the compositional data and gets you an association, and then it generates a network which we then again bootstrap 1000 times and p-value corrected, and essentially got a co-occurrence network of this microbial population, where in this network each of your node is a microbial microbe, and their association you can think about probability value, which says how much probability that they will be co-occur in a particular sample. So for these three environments we created the network. As you can see, they are very different in size. So first thing we wanted to see, because as we use very strict cutoffs, we wanted to see how much of the data we actually retained in these networks. So you can see we plotted number of bacterial features that we fed into the G-CODA and other steps, and then what is the number of nodes and number of edges, and it is proportional to it. So showing that it's getting similar sort of things in the network, and we capture across more than 50% of the bacterial abundances in these networks. Then we try to identify the communities in these networks and uses a unsupervised method, and it's identify the dense subgraph. And very interestingly, so we identified 3 clusters in the hospital, 5 in the metasub, and 2 in the offices. And as you can see here, office is coming close to metasub, all like some of the places, and in the other half of it you can see the hospital is coming close to metasub. So probably there is a chance or hypothesis that we have the metasub or metro station, being a public transport space, can act as a transmission mode or medium from different environments across the different places, built environments. And then we move forward seeing that, so these are the bacterial features we gave, and you can see there are certain good amount of overlap across these environments. But when we see the ages in this environment, you can see very less similar ages across this environment, showing that the microbes present in this environment may be similar, but their interactions are not that similar as such. So to go deeper into that, we identify if some age, so we segregated the age, if some age is particularly present in multiple environment, that's a generalized age, and if a age is very specific to a environment, then we segregate them into 3. First is if the nodes, on the basis of node, if the particular nodes are present unique to this environment, so it's called a specialized age with specialized node, then one specialized node, and then generalized node. Specifically, these 2 nodes may present in other environment but not interacting in this particular environment. And their distribution, as you can see here, so hospital environment, you can see a lot of unique ages, number of unique ages are very high there, whereas metasub and office also have very, around 20% of the ages which are generalized. But very interestingly, if you see on the hospital and metasub, there are a lot of ages which basically our third category, specialized, this category where we have specialized with both generalized node. That means those microbes present in other environments but not interacting in those environments. So there is a chance of that environment play a unique role in these interactions. Similarly, at class level, so this is at a certain higher level we are trying to see the interactions, so that was very specific age. So at class level interactions, you can see here in the hospital, so certain things I just wanted to point out. So alpha proteobacteria and other things which are very unique, like very much common in other places, but hospital has some Clostridia or some Clostridia corynebacteria negativicutes are very unique for the hospital. But if you see the gamma proteobacteria, which I said already that it harbors a lot of nosocomial pathogens, it has a lot of interactions here across gamma proteobacteria. You can see gamma proteobacteria also interacts here, but bacilli gamma proteobacteria being present here are not interacting among themselves but interacting with others. So certain kind of these things again ensures us that the environment play a very essential role in understanding this microbial interaction, and like saying that these microbial interactions are very unique to this environments. Then we try to see, okay, these are our observations from different abundance value, but it's just an observation. There is no biology as such included in this data as of now. So we try to see if we put some components of biology in it, how will that look like? Will their pattern will be consistent or something like that? So for that, all the microbes we got, we collected their genomes. If they're not available, we took the type strains, we annotated the genome, we constructed their genome scale metabolic models, which essentially captures a metabolic landscape of a particular organisms in terms of metabolites, in terms of reactions, and then we simulated those microbial interactions. And how we simulated, we constructed different communities, and as I said, let's say this is our graph, and then we determine different community size, and say 2 member, 5 member, and so given a community size, we picked a particular subgraph of this network, which has to be definitely connected, and they have to be present in at least one sample together to say that they are co-occurring. So for that, we identify different community sizes from this network, and to show that they are not happening out of any random phenomena, we picked same size of random community for given all the bacterial species. And from there we did 10 iterations, because our networks are very small, so at least office network was very small, so if doing more iterations may cause repetition. So then we calculate a score called Smetana. We utilized a fantastic tool called Smetana. So what Smetana does, Smetana calculates two sort of indices. One is metabolic interaction potential, another one is metabolic resource overlap. So metabolic interaction potential, if you see M minus I, M is basically number of metabolite a particular organism require when it is in isolation or in the same community. And when it is with the different community members, what it is required, number of minimum metabolite for its growth. And then if you minus it, you will get how much a particular organism is dependent on its community. And on the other hand, if you see this MRO score, which is a competition index, because it's essentially, if you see this part, it shows that what are the intersection of the particular metabolites in the particular metabolic potential of these organisms. So if they have a lot of more particular metabolite overlap, so they tend to compete for that particular resource and then cause a competition. So these are the two index we utilized here, and then we plotted them for the communities we discussed. So as in community size 2, we are not seeing nothing, so it's just everything everywhere. But as we increase community size 5, they started polarizing across two sides, and at community size 8, you can clearly see office, which is, so this is our MIP MRO axis, so something here should be more cooperative, something this side is more competitive, and somewhere in between something is a mix of community. So as you can see, with the higher community size, we can see that office is more competitive in nature, and then hospital you can see more cooperative. Random communities all over the places, and metasub is somewhere in between lying here. So that shows that hospital setups are more cooperative. There are certain hypotheses for that as well. So hospitals has a lot more human associated microbe, and they also tend to have more cooperative in the nature, and they have lesser number of usually genome size. Whereas any built environment microbiomes are usually, there will be a lot of stresses, so they try to be more in genome size, they comes with more genomic more metabolic potential, and then they tend to compete for certain metabolites or certain resources. And whereas, so there is another important thing that we are not seeing. So if we believe that if we increase community size more, then we will see, you know, some more sort of distinction, proper distinction we can see here. So to conclude this talk, so we used nearly 600 shotgun metagenomic samples from different built environment and showed that microbial communities are very diverse and distinct. And then a huge fraction of hospital microbes are associated with nosocomial pathogen and responsible for all healthcare associated infections. Built environment harbors a unique microbial interactions that they are very different across different species. And then hospital setups are more cooperative due to presence of variety of organisms. We see in the very first slide that they are very high diversity. And office on the other hand is very competitive because of very less diversity of microbial communities. And then as a future work, we are trying to address a few things like how is there a chance of microbe that is going out of hospital, can it sustain in the other communities like metro station and office? But that's what we are looking forward to do. And then by the end, I would like to thank my supervisor, Professor Karthik Raman, and Vijay, who is a BS Data Science student at IIT Madras, worked with me in this project. I would like to thank my funding from PMRF, and this is my lab group. So thank you.
Thank you, Pratyay. So we will take up a couple of questions.
Hi, so my question is like, how do you make the genome scale metabolic model specific to an environment? How do you integrate the constraints to make it specific to an environment?
Yes, so as of now in this model we haven't given any constraints. We consider that they are a sufficient amount of metabolites, but as you said that is possible. So we for other studies we use this approach. So let's say we identify minimal metabolites, say we give a say what is their wild type growth rate, and we put a very lower, say 5% of this is, let's say that is a minimal growth for this, and what all metabolites it's required. And then we put some bounds to that metabolite. Let's say there are glucose and something is only minimal metabolite required for it, and then we put a bound of say 10 that's long unit glucose per millimolar for dry weight or something like that. So we put constants on them, so that actually ensures that the organism is just thriving there. So that you can use, and then after putting the constant you can do similar kind of analysis to show if there are any particular environment associated effects or not. But as of now in these models we haven't used any
But like temperature and pH is a crucial factor which will affect the the behavior of the microbes. So how do you overcome that limitation?
Yes, so that's a drawback of this kind of modeling. So we are using genome scale modeling. It doesn't encounter any sort of environmental feature. It's just about metabolism or metabolic capability of a particular organisms. So yeah, that's the point. But we don't and we even so I got the data from a particular study. So we don't have even the information for the particular, and it's not actually a time point, it's various time points. So I don't have the information as well, but that's
no
good.
problem. Thanks.
Okay, thanks.
Hello.
Hi.
We are doing a clinical trial at Tata Memorial Hospital in Bombay, where we are doing gut microbiota, changes post antibiotic treatment. My question is, we are using inverse Simpson index as a parameter to decide microbial communities changes across time points of a patient. So do you recommend that inverse Simpson index, or your profiling of doing longitudinal analysis through different time points like you are doing in that?
Okay, so you're talking about how diverse is the community, you are using inverse Simpson or something. So there are multiple that kind of metric. I would suggest you try with all the metrics. There are certain metrics which are very specific. So I know certain metrics which actually focuses on, they are rare. So all these metrics are basically for, our abundant microbes. There are certain metrics which also focus on rare microbe by like checking the distribution as well. So I would recommend you to check across different metrics, and if you are it is consistent across these things, then only you should like go or just check the fundamentals if that is satisfying your goal.
Okay, thank you.
Hello, I have one question.
Oh, hi.
So very nice talk.
Thanks.
My question is that using this approach, can you pinpoint on keystone species in that microbial network or microbial community?
Yes, yes. So I had a slide on hubs. I am not. I didn't show that. So yes. So we can identify. So mainly we. I'll just maybe show you. Yeah. So I had this slide. I guess if I enter full screen, this. Okay. Anyways, you can see it, right? So these are the different microbial communities in the hubs, and we checked across different centralities to see. And another quick observation is you can see Staphylococcus aureus known pathogen is degree across the hospitals. Yeah, that way you can find what are a key contributor from the network to that particular environment, and how these are associating in other with the others of the organisms. You can identify that using different network parameters.
So very nice talk. I have a question regarding whether you have considered non-human based transmission of microbes between different niches or different regions.
Okay.
Avian, like birds can actually probably eat some waste and then transfer that microbe or a community subset of the microbe from one environment to another. So that might contribute to the enrichment. Like you said, for metro stations, because this human communication hub, there is very enriched microbial community. So it might also happen for non-human type of animal transmission.
Yes, so we checked only human host as of now, and as this is preliminary focused on hospital microbiome, so most of it are human microbiome. But yeah, that's a good session. We can go back and check about other host of these organisms. I really didn't see the host of these organisms, but maybe at the metro station that you said, possibly there is a host could be something else as well. But I checked only human, and most of these were human associated microbes. Yeah. Thanks. I look forward to sessions also as well.
Thanks a lot for a wonderful presentation.
Yeah.
How genome sequencing will influence microbes in the hub and as well as in the communities as such?
Genome sequences?
Genome sequencing will influence the microbes.
No, it's a metagenomic sequences, so it is what it is, but then metagenomics also come with some restrictions. So we have over the years seen that rare microbes are not captured by this like shotgun metagenomic sequences as well, but they are present, they are persisting there. So as of now, that's a restriction of genome sequencing, but again, with certain cutoffs, we always try to talk about what is prevalent and abundant microbes. Probably they play definitely a significant role, but then we tend to overlook the whatever is the rare microbiome. So genome sequence as such is so it's a metagenomic. We got all the DNA, we mapped it to reference sequences. We have a 600 GB zip database, and that's still huge, and then so it's whatever it is we are seeing there, whatever mapped, we move forward with that.
Thank you.
Hi Pratyay,
Hi.
Nice talk. Nice to meet you again. Can
Thanks.
you please go to slide 15?
Slide 15. Yeah.
When you increase the community size, I see that office and metasub is kind of clustered together, but random and hospital is somewhat similar. How do you differentiate between them, and then how do you base your inference based on this data?
Okay, so we tried calculating each axis. So we did a statistical test based on the p-values on MRO and MIP, but definitely if we can increase the community size, the problem here is if you see the office environment is very low. We have a good amount of data from metasub and hospital, but office has very low amount of data. So we cannot go beyond 8-member community because there are 14 or 15 members in the in the whole space in the network nodes. So there will be repetitions in the things. So I believe if we can increase community size, we will see more differentiations. That has already shown similar kind of things as already shown in different papers.
but still you are you able to right now say there is difference between random and I guess the hospital data?
So the plot is not here. So what we did, we project them onto MRO axis and MIP axis, and then we check their p-values. So on MRO axis it was not very different significantly, but on MIP axis it was very different. So MIP wise it was very different.
Okay.
Yeah.
Thank you.
Okay, so let's thank the speaker. And so we have the last talk of this session by Garima Arora from Translational Health Science and Technology Institute. So she will be talking about development of metabolic models for drug repurposing, a novel approach for precision therapeutics.